Carbohydrates.
There are four groups of carbohydrates, which is the first one is the monosaccharides. It is the simplest group of carbohydrates, and it is also known as simple sugars. Example, the most common monosaccharides are glucose, fructose, and galactose. There are other examples of monosaccharides, of which we are going to look at later in this chapter and also in chapter 5. Okay. And then the second group of carbohydrates is the disaccharides, which consists of two monosaccharides that are linked by a covalent bond. This covalent bond that links the two monosaccharides together are known as glycosidic glycosidic linkage. Okay, linkage. So there are several types of glycosidic linkage based on the position of carbon atoms that link the two monosaccharides together, which is there are 1,4 glycosidic linkage, 1,2 glycosidic linkage, and also 1,6 glycosidic linkage. Later we are going to look at where can we find these different types of linkage. And then the third group of carbohydrates is the oligosaccharides, which consists of 3 to 14 monosaccharides that are linked together. For example, oligosaccharides can be found attached to proteins that are embedded in the plasma membrane, and it is known as glycoproteins. And also there are oligosaccharides that are attached to lipids in the plasma membrane. We call it as glycolipids. So these glycolipids and glycoproteins, they have specific function in the plasma membrane, of which we are going to look at later in chapter 3, plasma membrane transport. And then the fourth group of carbohydrates is the polysaccharides. So polysaccharides are polymers composed of many sugar monomers. For example, we have starch, glycogen, cellulose, chitin, and murine. Starch and glycogen are storage carbohydrate. Starch are storage carbohydrate that are found in plants. They are usually stored in tubers, such as for example potatoes. And then glycogens are storage carbohydrate in animals, which are stored in the liver. And then cellulose is the um, component of plant cell wall, for example. And then you have murine and also chitin. Okay, so murine is the component of fungi cell wall, and as for chitin, it is the component of exoskeleton of insects. Okay. Okay, so we are going to look at the first one, which is the monosaccharides. So monosaccharides have molecular formulas that are usually multiple of CH2O. For example, the molecular formula for glucose will be C6H12O6. Okay, so that is the most common monosaccharides, and everybody knows the molecular formula for glucose. And then the monosaccharides are classified based on the position of the carbonyl groups. Okay, as you know, carbonyl group is carbon double bonded with oxygen. Okay, so based on the location of the carbonyl group in the monosaccharides, we can classify monosaccharide as either aldose or ketose. Okay, and also we can also classify monosaccharides based on the number of carbons in the carbon skeleton. For example, glucose have 6 carbons, so glucose is grouped as under the hexose sugar. Okay, under hexose sugar. Okay, so let's look at the next slide. Okay, so you can classify the monosaccharides based on the position of the carbonyl group. Okay, so if the carbonyl group is located at carbon number 1, as you can see here, carbon number 1, so the sugar are known as aldose or aldehyde sugar. So for example, we have glyceraldehyde, ribose, and also glucose and galactose. So these are monosaccharides where the carbonyl group are located at carbon number 1, aldose, okay, or aldehyde sugar. And then the other group is ketose or ketone sugar where the carbonyl group is located in the middle of the molecule, or usually at carbon number 2, okay, carbon number 2. Okay, so these are ketose sugar, for example dihydroxyacetone, ribulose and fructose. So glyceraldehyde and dihydroxyacetone, because they have 3 carbon atom in the sugar molecule, so they are known as trioses. Okay, trioses. And then for ribose and ribulose, because they have 5 carbon sugars, they have 5 carbon in the sugar molecule. 1, 2, 3, 4. What's the other one? 1, 2, 3, 4, 5. Okay, and then 1, 2, 3, 4 and 5. So these are known as pentoses, which is they are 5 carbon sugars, and the molecular formula will be C5H10O5. For trioses is C3H6O3. And then the last one is hexoses, where they have 6 carbon in the sugar molecule. So we have glucose, galactose and fructose. Okay, glucose, galactose and fructose, as we know, it is the most common monosaccharides. And here we have glyceraldehyde, dihydroxyacetone, ribose and ribulose, which I suppose it is new. Okay, so glyceraldehyde and also dihydroxyacetone, sugar molecule, for example, that we are going to come across in chapter 5, which is the substrate that are formed during cellular respiration pathway. And then for ribose and ribulose, ribose is the 5 carbon sugar that are found in RNA. Okay, in RNA. Okay. Okay, so these are the properties of monosaccharides. They are sweet taste. For example, your table sugar, which are commonly produced from sugar cane. So they have a sweet taste, and then they can be crystallized, and then they are polar. So it means that if the sugar are polar, they can easily dissolve in water. Okay, they can easily dissolve in water because they can form hydrogen bonds with water molecule due to the presence of hydroxyl group, which are attached to the carbon atom of the sugar molecule. Okay, so for example here, if I draw the structure of a glucose molecule, okay, so you should know how to draw the structure of a glucose molecule. So this is roughly a sketch of the sugar molecule glucose, CH2OH. Okay, so this is carbon number 1, carbon number 2, number 3, number 4, number 5 and number 6. Alright, and then at carbon number 1, the hydroxy group is here. Alright, and then the hydrogen atoms, which are attached to the carbon atom also. Okay, so due to the presence of many hydroxy group here and here and here also here, alright, so this will cause the sugar molecule that are easily dissolved in water, because they can form hydrogen bonds with water molecule. Okay, and then so they are easily dissolved in water, and they have reducing properties. What does this mean? It means that if you have a food sample, and then you want to test whether that food sample contains sugar or not, you will test the food sample with a Benedict reagent. Okay, so you will mix the food sample with the Benedict reagent, and then you are going to mix these two mixture together, and then you are going to heat in boiling water for several minutes. And if the color of the mixture changes to a range of color, for example, either the color changes to green, yellow, orange or brick reds, it indicates there's presence of reducing sugar. Okay, So the different color indicates different concentration of the sugar that are present in the food sample. Okay, so the next slide here, it shows you the ring form of glucose, and this glucose can exist in two forms as either alpha glucose or beta glucose. Okay, so for alpha glucose, the position of hydroxy group at carbon number 1 is located below the plane of the ring. Okay, carbon number 1, the hydroxy group is located below the plane, dekat bawah ya. And then the second type of glucose is beta glucose, where the position of hydroxy group at carbon number 1 here is located above the plane of the ring. Okay, so you should know the difference between alpha glucose and beta glucose, right? Okay, so monosaccharides are usually drawn as the linear skeletons. However, in aqueous solution, for example, in a cell, okay, so these monosaccharides form ring structures which is more stable. As you can see here, this is the linear skeleton for glucose. Okay, like we have looked in the previous slides, where glucose have 6 carbon. Okay, and the carbonyl group is located at carbon number 1. And then from the linear structure, you have to convert into ring structures if it exists in aqueous solution. So you should be able to know and identify the ring structure for glucose. Okay, for example, here if I were to draw a glucose molecule, so you just draw an oxygen atom here, and then you just draw a hexagon shape structure. Okay, and then as you know, glucose is a 6 carbon sugar, so you should be able to label all the carbons. So carbon number 1, number 2, number 3, number 4, number 5, and number 6. Okay, and then you just put a straight line at each edge here, indicating the presence of a carbon at atoms. Okay, and then you just add the atoms that are attached to each of the carbons. Okay, for example, at carbon number 2, the hydroxy group is located below the plane of carbon number 1. Okay, hydroxy group at carbon number 2, hydroxy group at carbon number 3, hydroxy group at carbon number 4, and then at carbon number 5 here you have CH2OH, and then the rest you just add hydrogen atoms. Okay, so this is how you draw a glucose molecule. Specifically, this one is alpha glucose. Okay, because hydroxy group is located below the plane. If you were to draw a beta glucose, okay, you just change the position of the hydroxy group, where the OH is located above the plane of carbon number 1, hydrogen is at the below carbon number 1. So this will become beta glucose. Okay, beta glucose. So you should know the difference between alpha and beta glucose. Okay, so next slide here is we are going to move into the next group of carbohydrates, which is the disaccharides. It is formed when a dehydration or condensation reaction joins two monosaccharides together. Okay, so as we have learned earlier, to join monomers together into a polymer or a disaccharide in this case, it is through the reaction dehydration, where in the reaction it involves the removal of a water molecule. Okay, so the bonds that form between the two monosaccharides to form the disaccharides, we call it as the glycosidic linkage. So there are 1,2 glycosidic linkage, 1,4 glycosidic linkage, and 1,6 glycosidic linkage. But in the disaccharides, the bonds is 1,4 glycosidic linkage. the linkage that forms between the two monosaccharides. Okay, later we are going to look at how is the linkage formed between carbon number 1 and carbon number 4 of the two monosaccharides. And the characteristics of a disaccharides is that it is water soluble, again due to the presence of many hydroxy group that can form a hydrogen bond, okay, hydrogen bonds with water molecule, causing the disaccharide to be able to dissolve in water, and then they have a sweet taste, and also they can be crystallized. Okay, so these are the three examples of disaccharides. We have the first one is the maltose or malt sugar, and then the monomers for the maltose is glucose and glucose. Okay, glucose and glucose. So so the bond will be alpha 1,4 glycosidic linkage, okay, that forms between the glucose and glucose molecule to form the maltose. And then the malt sugar is extracted, for example, or can be found in germinating grains, okay. And then this maltose sugar can function as respiratory substrate. What does it mean? It means that the maltose can be source of fuel to feed cellular respiration, for the cellular respiration to use to synthesize ATP, which is energy currency for cell to do cellular works. Okay, to form the aim is to form ATP, for example. Okay, respiratory substrate. So maltose can be used in cellular respiration to generate energy. Okay, and then maltose is a reducing sugar, so it means that when we react with Benedict reagent, it changes color. Okay, it changes color from blue to a range of colors from green, yellow, orange, and brick reds, indicating the presence of reducing sugar, indicating that the food sample contain maltose. Okay, and then the second example of the disaccharide is sucrose, which is cane sugar, which is the monomers is composed of glucose and fructose. Okay, glucose and fructose. So the linkage that links between glucose and fructose is called as 1,2 glycosidic linkage. And then the sucrose can be extracted or synthesized, extracted from sugar cane and also beetroot. Okay, beetroot, it is red in color. You can search if you don't know how a beetroot looks like, so you can Google to find out how a beetroot looks like. And then these are the source to produce your table sugar, the daily sugar that you used. Okay, and then it is the main form of carbohydrates that is transported in plants. Okay, so it is a non-reducing sugar, so it means that when you mix a sample containing sucrose with Benedict reagent, it won't change color. The color will remain blue. Okay, and then the third example of a disaccharides is maltose, so which is milk sugar, that can be found in milk. And then the monomers are glucose and also galactose. Okay, they can be found in milks from mammals, and then obviously it is a source of energy. Okay, that can be used during cellular respiration to generate energy in the form of ATP, and then lactose is a form of reducing sugar. Okay, alright. In this slide, we are going to look at the reaction, the dehydration reaction in the formation or in the synthesis of maltose, sucrose, and also lactose, the three disaccharides. Okay, so in the formation of a maltose sugar, so it forms between two glucose molecule and involves the formation of a water molecule, and the bonds that links between the glucose molecule, the glucose monomers of maltose, is we call it as 1,4 glycosidic linkage because it links between carbon number 1 and also carbon number 4. Okay, for sucrose, the monomers are glucose and also fructose. Okay, so the reaction also involves the removal of a water molecule, and it forms sucrose, and the bond that links between glucose and fructose, we call it as 1,2 glycosidic linkage, because it links between carbon number 1 and carbon number 2 of a fructose molecule. And then for the formation of lactose, the monomers are galactose and also glucose. And then again, because it involves the removal of a water molecule, hydroxyl, and also hydrogen atom, a water molecule is removed. So the bond forms between carbon number 1 and also carbon number 4 of glucose and galactose molecule. So the reaction is called as dehydration reaction or condensation reaction, because it removes one water molecule between the two monomers. Okay, so now we are going to look at how to draw the reaction. Okay, the dehydration reaction. Reaction for maltose. Okay, alright, so you know the maltose, the monomers consist of a glucose, two glucose molecule. So you draw the first glucose, oxygen, and then you draw a hexagon shaped structure. Okay, at each corner you draw a straight line. Okay, and then you label the carbon atom, number 1, number 2, number 3, number 4, number 5, number 6, and then the atoms that attach to each of the carbon. Okay, hydrogen, hydroxyl, 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 and then you here carbon number 6 is CH2OH, and then the rest you add hydrogen atoms. Okay, so this is the first glucose, and then you add with another glucose molecule. Okay, for each corner you add a straight line, CH2OH, okay, hydroxyl, 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 and then the hydroxyl, the rest is hydrogen atoms. Okay, so now you label the carbon atoms, carbon number 1, number 2, number 3, number 4, number 5, and number 6. Okay, in the process of dehydration reaction, a water molecule is removed. Okay, so this is the second glucose. Okay, glucose. Okay, so for the first glucose here, at carbon number 1, you remove a hydroxyl group, and for the second glucose molecule at carbon number 4, you remove a hydrogen atom. Okay, so these two molecules will combine together to form a water molecule that will be discarded from the reaction, removed from the reaction. Okay, so next, okay, because you have removed the hydroxyl from carbon number 1, the hydrogen atom from carbon number 4, so now you join the two glucose molecule together. Remember, there's still oxygen at carbon number 4. Alright, so this one you have to be very careful, okay, to draw the product, which is the maltose. Okay, so you just copy again the structure, oxygen, and then the hexagon shaped structure, and then beside it is the same hexagon, the oxygen, and then the hexagon structure, and then you put a straight line at each corner. Okay, so let me just number the molecule of, okay, 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, and 6. Okay, you draw a straight line at each of the corner. Okay, remember at carbon number 1 and number 4 you remove atoms, right? Okay, so carbon number 4 you just draw a straight line there, and also carbon number 1, and then you draw a line like that, and then oxygen at the middle, because you did not remove an oxygen atom from carbon number 4, so the bonds between carbon number 1 and number 4, there is an oxygen atom. Okay, and then the rest you just complete the structure. CH2OH H, okay, CH2OH, hydrogen, hydroxyl, 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 hydrogen, okay. So now you can observe that between carbon number 1 of the first glucose molecule and carbon number 4 of the second glucose molecule, there is a linkage that links between the two glucose molecule. So this is the a covalent bonds that form between the glucose molecule. Okay, so this covalent bond here we call it as 1,4 glycosidic linkage. Okay, glycosidic linkage, 1,4 glycosidic linkage between, because it links between carbon number 1 and also carbon number 4. To be more specific, you can also write alpha 1,4 glycosidic linkage because it forms between alpha glucose and alpha glucose monomers. Okay, so this is how you draw the reaction that forms maltose through the dehydration reaction. Okay. Okay, so now we are going to look at the dehydration reaction that forms for the second disaccharide, which is sucrose. Okay, so again you have to know the monomers for sucrose is glucose and fructose. Alright, so this one is a bit different where, okay, so for glucose you draw it as usual. Okay, oxygen and a hexagon shaped structure. Okay, you draw a straight line at each corner. Right, so you number the carbon atom 1, 2, 3, 4, 5 and 6. You put all the atoms, okay, for each of the carbon. Okay, hydrogen, hydroxyl, 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 and then the other hydroxyl, CH2OH. Right, okay, so for fructose you have to take note that this although fructose is a 6 carbon sugar, but the shape is like a pentose, but it is actually a hexose, a 6 carbon sugar, where let me just draw the structure. Okay, fructose. Alright, so oxygen is here, and then you draw a pentagon shaped structure. Alright, and then at each corner you draw a straight line. Okay, you draw a straight line. Okay, so now you number the carbon atoms. Okay, for fructose it is a bit different where the carbon number 1 is here. Carbon number 1, number 2, number 3, number 4, number 5, number 5 is here, and then number 6 is here. Okay, carbon number 1, number 2, number 3, number 4, number 5, number 6. Okay, now you complete all the atoms that attach to each of the carbon. So carbon number 1 is CH2OH. Carbon number 6 is also CH2OH. Okay, and then for the rest of the atoms at carbon number 2, there's hydroxyl. Alright, and then carbon number 3, hydroxyl. Carbon number 4, and then carbon and then and the rest is hydrogen atoms. Okay, hydrogen atoms. Okay, so the bonds that form between fructose and glucose is between carbon number 1 of glucose and carbon number 2 of fructose. So you will need to remove, okay, you will need to remove hydroxyl from carbon number 1 and then hydrogen atom from carbon number 2 of fructose to form a water molecule. And then you just join the two monomers together. Okay, you just draw it again for glucose and then for fructose. Okay, so each corner you just draw a straight line. Okay, so remember between at carbon number 2 of fructose, there is still oxygen. So you draw an oxygen atom at the middle here. Okay, then let me just number all the carbon: number 1, number 2, number 3, number 4, number 5, number 6, number 1, 2, 3, 4, 5, and 6. Okay. So you just have to know the placement of the atoms. Okay. CH2OH, CH2OH. Alright. And then hydrogen. Sorry, this one is not. Carbon number 6. OH, H. You just copy back again from the monomers. Okay, so now you have the complete structure of a sucrose. Okay, so as you can see, the bond that links between the two monomers, it links between carbon number 1 of glucose and carbon number 2 of fructose initially, and then so that means the bond that links between the two monomers is called as 1,2 glycosidic linkage. Okay, 1,2 glycosidic linkage between carbon number 1 and number 2. Okay, so you just have a practice of how to draw the reaction from the monomers until it forms the disaccharide. The third group of carbohydrates is the oligosaccharides. Oligosaccharides consist of monosaccharides that are linked together to form small chains, and it consists of about 3 to 14 monosaccharides that are linked together through glycosidic linkages. Oligosaccharides may be found attached to proteins and lipids. So if the oligosaccharides are attached to protein, we call it as glycoproteins, and if the oligosaccharides is attached to lipid, it forms glycolipids. For example, these two molecules can be found embedded in the plasma membrane of cell, and they have specific function which we are going to look at in detail in chapter 3. Okay, so the fourth group of carbohydrates is the polysaccharides. So polysaccharides are polymer of monosaccharides, and they have two main functions. They can function as storage carbohydrates such as starch and glycogen, or they can have structural roles such as cellulose, chitin, and MUREIN. So the structure and function of a polysaccharides are determined by its sugar monomers and the position of glycosidic linkage. So basically, the monomers for starch glycogen are composed of alpha glucose, and then the monomers between the alpha glucose molecule are linked together through 1,4 glycosidic linkage. The monomers of glucose in a straight
in a linear chain. Okay. And then if there's branches, the branch are linked together through 1,6 glycosidic linkage. And as for cellulose, the monomer is beta glucose. Okay, so cellulose is the component of plant cell wall, and as for chitin, it is the 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 component of it forms the exoskeleton of insects, and then you have the murine, which form the cell wall of fungi. Alright, so usually polysaccharides are insoluble in water due to their large structure, okay, and it is not sweet in taste. For example, starch, which can be found in potatoes, usually if you eat potatoes, it is not sweet compared to the table sugar that you usually add in your drinks, okay. And then so they are not sweet in tastes, and then important as food storage. Okay, food storage, starch in plant, glycogen in animals, building materials for cell or the whole organism. Okay, the whole organism is the exoskeleton in insects, which composed of chitin. Cell wall is cellulose. Okay, for plant. Okay, so starch. Starch, the first example of polysaccharide, it is a storage polysaccharide in plants, consists of entirely of alpha glucose monomers. So this one you have to remember. So plant cells store starch mainly as granules within a specialized organelle called amyloplast. So amyloplast is an organelle that function to store starch in granules. Okay, so potatoes cells are very rich in amyloplast. So potatoes are tubers, okay, which are found in the soil, and it contains starch. Okay, so plant hydrolyzes the starch, releasing the glucose subunit. So glucose serves as cellular fuels to generate energy needed for cellular works. Okay, so other example of plant that stores starch is rice, for example. Okay, rice. So rice, potatoes, tubers. So these are um, plant structures that store starch, where it is the source of food for other animals. Okay, it is the source of food for other animals. So when the animal eat this potatoes or rice, it can give energy to that animal. So because why? Because the subunit of starch is glucose. So when starch enters into the digestive system of that animal, it is then broken down into its monomer for which is glucose, and then absorbed in the blood of the animal, so that the glucose can be used by cell to carry out cellular respiration, of which the cellular respiration is used to synthesize ATP, which is energy currency for cell to do cellular activities. Okay, cellular activities. So the detailed explanation on cellular respiration we are going to look at in chapter 5. Okay. So starch is made up of two components, which is amylose and amylopectin. Okay, so the first component of starch is amylose. So the structure of amylose, it is a linear unbranched polymer made up of alpha glucose, and the glucose monomers are linked together by alpha 1,4 glycosidic linkage between the alpha glucose, okay, along the linear structure of the amylose that are coiled together. The glucose molecule are linked by alpha 1,4 glycosidic linkage between carbon number 1 and carbon number 4 of adjacent glucose molecule, carbon number 1 and carbon number 4. And the amylose chains, although it is unbranched, but it is coiled into a helical structure. and this helical structure of the amylose are held together by hydrogen bonds between the hydroxyl group of the glucose subunit. uh, so so the glucose molecule, they have hydroxyl, they have a hydroxyl group. So the hydroxyl groups and the other hydroxyl group of another glucose molecule will form hydrogen bond to hold the helical structure. Okay, the helical structure are held by hydrogen bonds. So these are the structure of amylose. It is unbranched, it is coiled into helical, and the helical structure is held together by hydrogen bonds. The monomers of the glucose are held together by alpha 1,4 glycosidic linkage. The second component of starch is amylopectin. Okay, so amylopectin is different, is a bit different from amylose, where amylopectin, they have branch. Okay, they have branches. So each branch of amylopectin is a short chain made up of many alpha glucose. Again, the glucose molecule are held together by alpha 1,4 glycosidic linkage. So between the glucose molecules, it is alpha 1,4 glycosidic linkage, but the branches are held together by alpha 1,6 glycosidic linkage. It means that, okay, so here you have the main polymer, okay, within the amylopectin, and then where there's branch, the branches are held together by alpha 1,6 between carbon number 6 and carbon number 1 of the branch. Okay, so that is the structure of amylopectin. Where they have branch is 1,6, between the monomers is 1,4. Okay, so the next storage carbohydrates is the glycogen. So glycogen is the storage polysaccharides in animals. So human and other vertebrates stores glycogen mainly in the liver and also muscle cells. Okay, so when animals or human, they eat carbohydrates, for example, they eat potatoes or rice or bread, okay, whatever sources that contain initially starch from plant, for example. So this starch will be absorbed, will be digested, and then broken down into glucose, absorbed into the bloodstream, and then the excess glucose within the blood will be stored in the form of glycogen, and they are usually stored in the liver. Okay, they are usually stored in the liver or in the muscle cell. So the structure is similar to amylopectin, but it is a larger macromolecule made up of alpha glucose with more branches. So compared to amylopectin, glycogen will have more branch. Okay, they have more branches, and they are more water soluble. Okay, so this is the structure of a glycogen. So again, it is helical in shape. Okay, the bonds between the alpha glucose molecule is 1,4, where there's branch is 1,6. Okay, is 1,6. So where there's branches, so the bonds are 1,6. Between the alpha glucose is 1,4 glycosidic linkage. So glycogen, they have more branches compared to amylopectin. Okay, so the next type of polysaccharides is cellulose. So it is the major component of the tough wall of plant cell. Okay, so you have a plant cell, so you have the plasma membrane within, and then outside is protected by a cell wall. Okay, so cellulose is a polymer of beta glucose. So the monomers of cellulose is beta glucose, and the beta glucose are linked together by beta glycosidic linkage. So different from starch or glycogen, where the monomers are alpha glucose. So the bond is, we call it as alpha 1,4, for example, or alpha 1,6. But for cellulose, because the monomers are beta glucose, so the glycosidic linkage, we put beta in front. Okay, so beta glycosidic linkage. You can use both glycosidic linkage or glycosidic bond. Both will be accepted. Okay, so in this slide, it shows you the different between the formation of of bonds between the monomers that are found in starch and also in cellulose. For starch, because the monomers are made up of alpha glucose, so the alpha glucose monomers are in the same orientation to form the helical structure of amylose or amylopectin. So as you can see, so the, for starch, okay, the monomers are alpha glucose. Say, so this is the first glucose, second glucose, third glucose, fourth, and so on. So they are placed, the glucose are placed in the same orientation. So it means that, as you know, so this is carbon number 6, carbon number 6, carbon number 6, carbon number 6. So the alpha glucose is placed in the same orientation, okay, to form 1,4-glycosidic linkage between the monomers, okay, within the starch. But as for cellulose, okay, because it is made up of beta glucose as the monomers, so the beta glucose molecule are arranged upside down, okay, upside down with respect to its neighbor. Okay, for example, in this diagram, as you can see, so the first beta glucose is oriented in the upright position. Okay, so carbon number 1, number 2, number 3, number 4, number 5, number 6. But the beta glucose next to it, it is arranged in the upside down orientation. Okay, so as you can see here, this is carbon number 1, number 2, number 3, number 4, number 5, number 6. So as you can see, carbon number 6 of the first glucose is upward. It is placed upward, but the second glucose is at the bottom. Okay, number 6 up, number 6 at the bottom. So they are placed, the beta glucose are placed or arranged upside down with respect to its neighbor. The first glucose upright, the second glucose is placed upside down, upright, upside down, like that. Okay, but the bond is still between carbon number 1 and carbon number 4, carbon number 1 and number 4. Okay, the bond are called as beta 1,4-glycosidic linkage for cellulose. Alright, in this slide we are going to look at the arrangement of cellulose molecule that forms microfibrils. Okay, so as you can see here, the beta glucose molecules are attached together to form a linear cellulose molecule. So each of the cellulose molecule is a straight structures which are arranged parallel to one another. So this is one cellulose molecule. Adjacent to it, there's the next cellulose molecule, and then the next cellulose molecules. So these cellulose molecules are arranged parallel to one another. And then the adjacent cellulose molecules, the straight cellulose molecules, they are unbranched. They are held together by hydrogen bonds, okay, between hydroxy group of one glucose molecule and a hydroxy group of another hydroxy molecule of the cellulose molecule adjacent to it, parallel to it. Okay, so these parallel arrangement of cellulose molecule are held together by many hydrogen bonds between hydroxy group of glucose molecules. So this arrangement, the arrangement of parallel cellulose molecules, it will form microfibrils. Okay, so it forms cellulose microfibrils that forms the cell wall of plants. Okay, these are structural polysaccharide. It forms structures. Okay, so the next structural polysaccharides, or polysaccharides that has structural structural function, is chitin. Okay, chitin. So chitin is another structural polysaccharide. It forms structures, so which is found in the exoskeleton of insects, crayfish, and also arthropods, insects. Okay. So chitin can also provide structural support for the cell wall of many fungi. Okay. So if you look at this diagram, so this is the monomers. Okay, the monomers that form chitin. So if you look at the monomer, okay, the monomers is similar to the so the structure of beta glucose, except for at carbon number 1, beta glucose, because the carbon at the hydroxyl at carbon number 1 is located above the plane of carbon number 1, but at carbon number 2, instead of having hydroxyl, it is replaced with this nitrogen containing component or appendages, we call it. Okay, so that is the difference between the monomer of cellulose and also chitin. Okay, so chitin, the monomers contain nitrogen containing appendages at carbon number 2. Okay, so the next the next structure is murine. Okay, so murine, the next polysaccharides. Okay, the next polysaccharide is murine or peptidoglycan. So it is a polysaccharide. It is a component of bacterial cell wall. So forgive me, earlier in the video, murine is actually the component of bacterial cell wall. Okay, bacterial cell wall. So it consists of polysaccharides cross-linked with amino acids. Okay, so these are the polysaccharides. Okay, the polysaccharide chain that are linked together by amino acids. Okay, amino acids. Okay, amino acids. So these are amino acids. So that's it for this part. We are going to continue in the next video for the next subtopic, which is on lipid.